All right. So I'm going to give a brief overview here of the science of COVID vaccines. I'm not going to get into the moral and ethical issues yet. We'll just let Brett and Melissa will handle that after I've given this overview. But to understand that and put it in the proper context, it's good to have sort of an understanding of the science behind these vaccines, how they're developed, what the cell lines in particular, what they are, and how they're used in those. So in general, just to start, what do you want a vaccine to do? Because once you understand this, you can see why these are developed the way they are and what cell lines might be used for them. So a successful vaccine is going to generate antibodies and immune cells, particularly T cells, that are specific to the pathogen of interest. So in the case of SARS-CoV-2, you can see a diagram of that up in the top right here of this slide. And how the immune cells in your body recognize this, they recognize the protein sticking off its surface. So that's how they'll recognize that something is foreign and mount an immune response against it. In particular, what vaccines developers are focused on is this spike protein that you can see there in orange that's sticking off of the SARS-CoV-2 virus. Now, when the virus gets into cells, you can see down the bottom like a cartoon of a viral infected cell. When it gets into cells, what cells do, one of their natural defense mechanisms is they will take some of the virus proteins that the virus is making. So when the virus gets into cells, it starts making its proteins, particularly in this case, the spike protein, and it will advertise them on its surface. So the cells that are infected will display pieces of these proteins on those molecules you can see there in purple on their surface. They advertise that to the immune system, and then the immune system responds, hopefully by generating antibodies and T cells through a variety of complex mechanisms. The goal of the vaccine is to, one goal, a way to develop vaccines is to get the individual cells in your body to express these viral proteins without actually infecting the person with the viral pathogen. So how can you get your cells to display the spike protein to the immune system such that the immune system will generate antibodies and T cells against that without actually having to undergo the infection? Right? So there's two ways in which the currently approved vaccines do this. So we have 3 vaccines currently approved in the US. Now there's other vaccines that are under development that use different mechanisms. I'm not going to go into those, but let's focus on the 3 that are out there in the market right now. You have the Pfizer vaccine and the Moderna vaccine. Those are the 2 that were initially approved, and they were both made using synthetic messenger RNAs. And these are the first 2 messenger RNA based vaccines that have come to market. And so what they do, the way they work, is you see they have synthetic messenger RNA. So you synthesize a messenger RNA that corresponds to the spike protein. And once a messenger RNA is inside of a cell, the cell will read that and make the spike protein. So if you can get the spike protein messenger RNA into your cells, your body will start making the spike protein and advertise it to the immune system. And the immune system will recognize it and hopefully make antibodies and T cells against it. So you package the messenger RNA in a sort of a lipid vesicle, you inject it into the body. The RNA gets into your cells, they express the protein, and you make the antibodies and the T cells. But the person, the full virus never gets in. The person never, it comes down with the major symptoms of COVID-19, right? The Johnson & Johnson, the most recent one to be approved, uses a different mechanism, but it's a very similar process. Right? In this case, what it uses is an attenuated adenovirus. This is a human adenoviruses, they infect human cells, but it's attenuated, so it can't replicate. Right? And what they'll do is they'll modify it. So you take, you see in this figure here, that green line. So that is the gene, the DNA for the SARS-CoV-2 spike protein. And they genetically modified this virus and put it in there. So now you've got this adenovirus with the SARS-CoV-2 protein in it. And then if you inject that into an individual, the adenovirus can insert its DNA into your cells. But because it's defective, it's attenuated, it can't replicate. But now that SARS-CoV-2 gene is in there, the cell will produce the protein, express it on its surface, and activate the immune system, hopefully with those antibodies and T cells. And so again, you've got the cell to activate the immune system without actually infecting it with the SARS-CoV-2 virus. So these are the two ways right now, the vaccines that are out there on the market, the way that they work. Now, with understanding of how they work, let's look at the cell lines that are used, and we'll explain what these cell lines are, and then why they are needed for making the Johnson & Johnson vaccine, and why it's used not to make, but to test the other two, sort of quality control to make sure they're working. Okay? So the two key cell lines that are going to be that are being used for the development of COVID-19 vaccines are these HEK293 cell line and the PER-C6. Now, the vaccines that we just talked about that are on market, they both use the HEK293 cell line. So I'm just going to focus on that, just so you know there's other lines there's another line that's similar. Now, the HEK293 line was derived in the 1970s, and it's from an aborted fetal tissue, from kidney cells, from an aborted fetal system, and so it's a human embryonic kidney cell line. Now, this cell line has been modified so that it's immortal, so you can grow this cell line indefinitely in the lab. So you don't need to go back, once you have this cell line, you don't need new aborted fetal tissue to maintain it. It can be maintained sort of indefinitely. And it's now widely used in the research community and for drug and vaccine development. One of the reasons that it's useful is that it is able to support the growth of that attenuated adenovirus vector that we saw on the previous slide. And I'll get more into that on the next slide. Other reasons, it's very well established and characterized. So it's sort of like a gold standard in research. It's used widely. It's easy to use to compare. So if you test a drug in this cell line, you can compare it to data for somebody else that tested another drug on that same cell line. It's very easy to culture. It's immortal. You can grow it indefinitely. It's easy to modify, so you can get genes into it relatively easily if you want to modify it. It's very good if you want to express human proteins in it and sort of characterize them, like the spike protein, for example. Now, the key use of the HEK293 cells in the vaccines that are on the market right now is they are used to actually produce the Johnson & Johnson vaccine. So the way it's used here is you make the modified virus vector. So you insert the spike protein gene into that modified adenovirus, right? Now, what you need to make the vaccine is you need to get lots of that virus with that modified gene in. The problem is that it doesn't grow. It can't replicate in most cells. It's replication deficient. But the HEK2 cells have been modified so that they can replicate in there. So you can then infect the HEK293 cells, and the virus will grow really well in those. And then you can purify the virus and get rid of the cells and get large amounts of virus. So it's a good cell line for making large amounts of this viral product that then you can inject as part of the vaccine. So it's important to recognize that the HEK293 cells are not part of the final product. Okay, they are removed from the final product. The final product has the virus in it and not the actual cells that you've been culturing. Okay. Now, in addition to being used for making the Johnson & Johnson vaccine, I'm going to jump back, they're also used for quality control for these synthetic messenger RNA vaccines. So when you make the synthetic messenger RNA and you coat it in these lipid membranes, right, and you manufacture it, you have to always test each lot to make sure that the messenger RNA is still working. So they'll take a sample from that lot and they'll infect these HEK cells, make sure that the RNA gets into those cells, is stable, and produces that spike protein on the surface of those cells. And if it is, then okay, that batch is good. So it's used for this ongoing testing of these, but it's not actually needed to manufacture. This is totally synthetic. You don't need a cell line to make this messenger RNA and the lipids that it's surrounded in. Now, just to clarify, these HEK293 cells are used sort of ubiquitously in research because of all those properties that I talked about before. They're used widely in basic research, and you know, if you need to express a human protein to examine how it works, it's a very useful and easy cell line to do that in. So you can get to the final product a lot quicker with a lot fewer headaches than many other cell lines. So it's used for the testing of a lot of existing medications, for the development of new medications. As you can see, the development and manufacturing of vaccines, and also for the development of food additives. So it's widely used. And part of the reason it's widely used, like I said, it's a gold standard. There's so much literature out there. It's become sort of a standard sort of testing line. And it's also easier because it's so well characterized, it's also easier to get this through FDA approval. So if this cell line is being used in the manufacturing of a vaccine, because there's so much data about this cell line, it's easier to get the FDA approval rather than using some novel cell line that then you might have to spend much more time characterizing to get regulatory approval for using that cell line in sort of drug manufacturing. Now, the last thing I want to talk about before I hand this off to Melissa, I think will be next, is to just briefly look at the clinical trial data for these 3 vaccines and talk a little bit about the risks associated with these vaccines, because there's a lot of information and sometimes misinformation out there. And just to summarize, you see these 3 different vaccine trials here, and there are other trials of these in other countries. But the 3 that were used to get approval in the US, you can see that both the mRNA vaccines, which are the first 2, the Pfizer and the Moderna, had about 95% effectiveness in preventing symptomatic infection. The Johnson & Johnson was a little bit lower, but more importantly, what really matters is it was 85% effective in preventing severe infection and hospitalization and death. So they're all relatively equally good at preventing those severe infections, which is really what we're concerned about, preventing sort of mortality with these vaccines. Often that 65% number is sort of misinterpreted. Oh, it only works 65%. But it's very good at preventing severe infection. People that have that are more likely to develop a mild infection than someone that had the Moderna or Pfizer vaccine. In general, the safety data was quite good on these. You can see over 100,000 participants in all 3, if you add those up. And there's a lot of local immune reactions that occur when you get the injection, but what we're concerned about is sort of serious adverse events and deaths associated with taking these vaccines. And so what they found in all 3 trials is that there was no difference in the amount of serious adverse events you found in the days after being administered the vaccine in those who got the placebo versus those who got the control. There was no difference. And even in the Pfizer one, there was actually more deaths and serious adverse events in the control group. So relative to the control group, there seemed to be no sort of safety issues. Now, these data that was initially used was only 2 months of safety data. So is there long-term effects? That's one of the questions we don't really know. But that said, the US, we have the vaccine adverse event reporting system that's in effect. And so if there's ever an adverse event, it has to be reported. So there's ongoing safety monitoring. You may have heard there was one vaccine in Europe that they halted the administration of that. That was the AstraZeneca vaccine because they thought there was elevated incidence of blood clots for people that took that. But that was really halted out of an abundance of caution. If you look at the data, there was no difference in the number of blood clots you found in the individuals that got vaccinated than you would expect in the general population. So you can think if you vaccinate 1 million people and you follow them for a month, versus if you just follow some normal people for a month, there's going to be certain people that have blood clots that develop in both groups. And so there was, they did recognize that, but it wasn't an elevated risk. But there is ongoing long-term safety evaluation of these as we move forward. There also needs to be more study on pregnant women and on children. The Pfizer trial looked at 16 and older. Moderna and Johnson & Johnson only had 18 and older. So you're younger than 16, there really isn't good data. There is sort of caution, people that have immunosensitivity. There's particularly some of the additives in these vaccines, so like polyethylene glycol is added to these. And some people are at risk of anaphylactic shock from that, but they often have been sensitized from previous exposure. And that's really sort of the main sort of risk. And also those that have active COVID-19 infection shouldn't take the vaccines. Wait till that clears before they would take that. But in general, you know, the safety data looks relatively good in these. Now, you can say there isn't long-term safety data yet, but the Johnson & Johnson vaccine, there's many vaccines on the market that have been developed in the same way as the Johnson & Johnson vaccine with very good safety data long-term. Now, Pfizer and Moderna, there's a lot of concern because these are the first RNA vaccines, but you can see the mechanism between Johnson & Johnson and the Moderna and Pfizer is very, very similar. And actually, RNA is probably, it's less stable. So there's probably less long-term side effects because it probably gets degraded quicker than, say, the DNA that gets put in the Johnson & Johnson. So on first principles, you might expect fewer long-term side effects with Moderna and Pfizer than you would with Johnson & Johnson, which is using a methodology that's been well established in vaccine development. So I'll stop there and we'll turn it over to the next presenter.